So thank you, Adeline. I think that gives you a very nice summary of how we've interjected germline testing for mutations in the clinic. I'd like to take a couple of minutes and sort of review where we're at in terms of identifying the right therapy for the right patient by bringing in sequencing of the patient's tumor. And that's a program that Michelle Capabianco is supporting through Pancreatic Cancer Canada. So, you know, we have made progress in pancreas cancer. So this slide shows the progress over the last decade or so. And in fact, over the last 5 years, we've had progress made in terms of the types of chemotherapies we have. So what you could see is, you know, when I started training, or just after I started training, the 1-year survival if somebody got diagnosed with advanced pancreas cancer, these are patients that have disease that we could control, hopefully. The 1-year survival, the median, so the average 1-year survival was 2%, and we've increased that over time with new drugs to 48%. I know it's not great, but it's progress. So we're identifying agents. So this is a cocktail called FOLFIRINOX. This is another cocktail, gemcitabine, with something called Paxtel or Abraxane. We're finding that these cocktails do make a difference and do improve tumor responses. And we're also seeing that they improve patient survival. We just have to figure out how to get them to the right patient. And at the same time, there are studies that are coming out where we've improved on the types of chemotherapies that we have to give patients after surgery. So we know we need chemotherapy for patients that have advanced disease to control their disease, but we also know that we need chemotherapy as an adjunct to surgery to be able to provide the patient with the best chance for cure. So surgery plus chemotherapy equals the potential for cure. So we're finding that we're getting new trials that are identifying new drug cocktails that we could use after surgery. And here's something very encouraging. So this has not come out in a publication yet, so it's not in a journal, but it was presented at the largest North American clinical oncology meeting this past, it's called ASCO, this past May, June. And this was a study, again, Canadian contribution. This is a French-Canadian, CCTG is the Canadian Clinical Trials Group, and it's a French and Canadian contribution. And what we did in this study is we compared FOLFIRINOX versus the standard of care after surgery, which was gemcitabine. And what you could see here, the median survival is nearly 55 months, so the average survival, if you translate this, it's more than 4 years of survival. 5 years of survival is considered curing cancer. We're getting pretty close to it, so that's quite encouraging. So again, we could probably do better if we could figure out which chemotherapy to give to the right patient. And that has changed their practice. So if you have surgery at our hospital and most Canadian hospitals at this point, we try to give you this cocktail FOLFIRINOX after surgery to improve your chance of survival. So we can do better, and this cartoon, I think, says it all. One size does not fit all. And here's the hypothesis. You know, this is research. This is the innovation part. And the innovation part is if we profile the tumors of a patient, we take a biopsy and profile that tumor, maybe we could figure out what is the right chemotherapy for the patient. And that's where we're at. And this is where PANC-1 came in. So we had to build an infrastructure. So we had individual infrastructures at hospitals. I built my infrastructure by bringing in cancer genetics and genetic counselors into the clinic and building a program around that. And other institutes have done it differently. But we had to build some infrastructure, and we need our national organization. So this is not just about telling you about science and what progress we made in medicine, but the importance of philanthropy. And I think philanthropy has helped in many diseases, and it's really stepping up to the plate when it comes to pancreas cancer. Unfortunately, patients with pancreas cancer used to no longer die within a year for the most part. Their families are devastated. The last thing on their mind is to get up there and advocate for pancreas cancer and bring research dollars to pancreas cancer centers. So this is where Michelle Capobianco, Craig's Coss, and other Canadian organizations, but really under the championship of PCC, Pancreas Cancer Canada, have come together and have helped us put a network together across Canada. So we formed this PANC1 network, and this led to this, which was very exciting for us. It was one of the first profiling. There's been other profiling studies done, tumor profiling studies done, but we were able, this was a 3-year project to get the funding for this, and we used not only the seed funding from PCC to get this going and establish our program, but also as matching funds for this initiative. And there's also additional matching funds through the Ontario government. And what is this program? It's essentially a clinical trial where the patient comes in. So it's called EPIC, and EPIC is the national program, and across Canada we've divided it into Eastern Canada and Western Canada. There's a lot of acronyms in medicine and oncology, and it gets confusing, but for those of you in Eastern Canada, the program is called COMPASS, and here's what COMPASS stands for. So if you're a patient that comes and sees me in clinic today with a diagnosis of pancreas cancer, advanced pancreas cancer, inoperable pancreas cancer, and you are healthy enough that we could give you advanced chemotherapy, so modern chemotherapy, that FOLFIRINOX that I described, or the gemcitabine and Abraxane, you're going to get a biopsy within a week to 2, and your start chemotherapy within a week to 2. And that biopsy is then going to be taken, it's going to be processed, we're going to sequence all the DNA in the tumor, we're going to sequence the RNA in the tumor, and we're going to have those results ready for you within 8 weeks. That means we have to turn this around in 8 weeks and have a molecular tumor board discussion, so we have to sit and discuss it, and then be able to provide information at 8 weeks to your treating physician more than to you, because the treating physician will be able to interpret it and share it with you, so that if your first-line chemotherapy fails, there's opportunity to intervene and give you a second-line chemotherapy that's ciblé, or targeted therapy. So that's the goal of this program. So that's what we do. So we have the patients. We have what's called a molecular tumor board. In Eastern Canada, we do it every 4 to 6 weeks. We have to have enough patients to discuss. It's done by teleconference, and it's between Toronto, Montreal, and Kingston has joined in. I think the SHIM will eventually join in as well once the study's open there. Ottawa is about to open as well. So the more minds we put to task, the better results we have, and the more ability we have to identify targeted therapies. So one would say, so here's more progress, one would say in a disease that's so devastating, most patients would not benefit from second line. Well, the fact is that 34% of our 190 somewhat patients that have been on the study so far, so the Eastern Canada study, have moved on to second line therapy. So clinical trial is the best care for patients with difficult to treat disease. It also means that these patients are monitored and watched carefully, and their chances to move on to a second line therapy increases as well. So that's another point of progress. So we talked about BRCA causing cancer. This is a subtype of pancreas cancer that is the first meeting we had, the first public forum we had was with BRCA, and Libby Zyman came in from Toronto. She inspired me to do personalized oncology medicine as a patient because she's of Ashkenazi Jewish descent, Montreal roots, by the way, and she was diagnosed with locally advanced pancreas cancer several years ago, many years ago. She's cured now. She had a BRCA mutation because of her genes. And she responded to targeted chemotherapy, so such as we thought at the time. And then she went on to have her tumor taken out, and she's cured, and she's a quite vibrant and active member of the Canadian community and has advocated for pancreas cancer. So that inspired me, and what I've learned over time through the research we've done at the Goodman and elsewhere is that this subtype of pancreas cancer represents up to 10% of all pancreas cancer. Important because we understand it, and maybe we could make therapeutic differences. So it's not important, it's simply to show you that out of the 290 genomes that we have sequenced, so tumors that we have sequenced, their genome, their whole DNA looks something like this. And if we look at so we have one of the largest collections, and now they've increased in size, of tumors of patients that have a BRCA mutation in their blood, in their DNA, that they could pass on in families. Their DNA looks different, and you have to trust me on that. It does look different, and we're able to use this information to try to understand how to treat the patients. So I can't go off in 2018 and not touch on immunotherapy. Everybody comes to clinic, asks me if we could give immunotherapy for pancreas cancer, and it's quite timely because this year's Nobel Prize in medicine was awarded to two folks that made contributions in the 90s to the field of immunotherapy, and that's quite remarkable. Most of the time, Nobel Prizes are awarded when many years later, rather than over this short period of time. And these contributions that these two folks made, one American, one Japanese, have changed many tumors, have changed how we treat several tumor sites. And one of the others, I have to put a plug in for my friend and colleague Ian Watson, one of the other leading efforts supported by the Terry Fox is a melanoma effort, looking at how we could identify, and that's being run out of the Goodman Cancer Center, using these advances. But in pancreas cancer, I'm not sure that immunotherapy is the answer. So if you look at the tumors that respond, so the tumors that respond to immunotherapy are the ones on this side of this graph, and the ones that don't respond are down here, and pancreas cancer is down there. But there are forms, so colon cancer also doesn't respond, but some forms of colon cancer respond. What's on this axis here, so the forms of colon cancer that respond are the ones that have high load of mutations, high number of changes in their DNA. Those are the tumors that will respond. So these are tumors that have mutations in some genes that are involved in repairing the DNA when there's damage to DNA as the DNA replicates as cells divide. And the same is true for other cancers that arise from these germline, these hereditary mutations in these DNA repair genes. And some of these non-colorectal cancers include pancreas cancer, but it's a very small fraction. So for a very low number of patients, maybe there's a benefit in immunotherapy. And again, another Canadian contribution is what's called the PA7 CCG, Canadian Clinical Trials Group trial, which is looking at immunotherapy in pancreas cancer. It was open at our center and several centers in Quebec, and it's now closed and the data is being evaluated. So what you could see from this slide here, this is looking at the number of mutations in pancreas cancer, and you could look at the number of mutations goes up to 80,000 here. If you look at these number of mutations in these patients that have this MMRD, which is mismatch repair deficiency, so something called Lynch syndrome or mutations in these DNA repair genes, their mutational load is here. So they need to have, these tumors have many errors in them. And as a result, they give rise to new proteins, new markers in the tumor, which immunotherapy could recognize and kill the tumor. So here's BRCA. So the average pancreas cancer will have a very low tumor value. BRCA is somewhere here. It's at the upper end. It doesn't quite approach these guys that respond to immunotherapy, but it's at the upper end. So that's where we're going next. This is just to remind me to tell you that if you look at the cancer cells of BRCA tumors, they look very different. They look like cancer cells that are being attacked by the immune system. And Dr. Morag Park in her breast cancer research has shown this quite nicely in breast cancer, and there may be some parallels with pancreas cancer and breast cancer. So that's what we've done with Compass, we're learning, we're identifying, we're trying to understand the disease, and we're trying to identify therapies for patients in second line, and more than a third of our patients go to second line. But it's really just designed for second line. And where we're going to make a difference is if we could take this information and bring it to first line. And that's what we're learning, and we're making observations that will help us design first line therapies. And I'm going to share with you in closing one important contribution that we've made, or making, or learning from Compass, and what our next steps are. I'm going to move along, and I want to come back to this analogy. This slide is taken from my class I'm giving next week, and it's different ways that we could characterize the tumor, pancreas cancer tumors, at a molecular level. And this is an important classification by this fellow called Moffitt. And it's called basal-like and classical-like. And somebody like Dr. Park right now should be excited because this sounds like breast cancer. And this is what breast cancer looks like. This is the first observations made by breast cancer. So Michelle said breast cancer, where it was 40 years ago, well, we're at least 20 years behind, no longer 40 years behind, and we're making those observations. So you could classify pancreas cancer into basal-like and to classical by looking at the RNA. And what's really cool about this is, remember we said there are two modern chemotherapies, FOLFIRINOX and the other one called gemcitabine and Abraxane. These guys in purple here are all patients in the Compass trial that have been treated with FOLFIRINOX and have had the classical type. Their tumors respond to FOLFIRINOX. Patients that have the basal-like that are given FOLFIRINOX don't respond. In fact, newer data suggests that it may be doing harm to them, to giving patients FOLFIRINOX if they have the basal-like. So this sort of summarizes what I just said in a different graph format. And we've taken this information and we're like, we could apply this clinically. Maybe we could use this information to decide what type of chemotherapy to give a patient. But it's not practical. It takes 8 weeks, remember, to sequence the DNA. It takes 8 weeks to sequence all of the RNA of the tumor. It takes a biopsy and so on. So we need to find something practical. So Sandra Fisher in Toronto is a talented pathologist, and she's identified. So from that classical and basal signature, we could pull out one gene, and we could see if that gene is a surrogate that could sort of help us characterize or put patients into one of two baskets, classical or basal. And this gene, we think, is GATA6. Doesn't matter what the gene does. It does matter. It's somebody's PhD thesis. But for the purpose of us identifying what to do with patients, it doesn't matter. What we need is a clinical assay. So here's the other part of innovation. So you make an observation. It's got to go through peer review. It's got to be published and accepted. And now you have to bring it into clinic. So where we're at now is trying to standardize this test, this GATA6 test, into the clinic so that we could bring it and put it into clinical trials. So that's where we're heading. That's what we want to do. What we want to do is use a test like this to identify which patient should get FOLFIRINOX, which patient should get Abraxane, because those are our two mainstream therapies that we're seeing reasonable results, and we think we could do better by personalizing the therapy. So I end with this circle of care. It's all what it's about. Patients in the middle, and everybody works for the patient. Thank you for your attention.